Okay, we will make a start now. So welcome everyone to our A Level AQA Biology session. If you haven't already, please do scan the QR code to confirm your attendance to the session. We are going to make a start now. Okay, so in today's lesson, we're going to be continuing from what we've already looked at in our last lesson. In our last lesson, we looked at DNA replication, and in today's lesson, we are going to be looking at the second part of DNA replication. It's a small part of the spec, but it's a very much ignored part of the spec that examiners love to ask questions on. Okay, and that's the Watson and Crick model of DNA replication. So what we're going to start off by looking at is basically the semi-conservative replication of DNA. And then we're going to move on to the Watson and Crick model by looking at an image which I have created. And then we're going to finish off by looking at some exam questions. Okay, if you have any questions at any point, please just do let me know at any point in the Q&A. I'll be more than happy to answer them for you. Okay, so this is the part of the specification that we are going to be focusing on. We're going to be evaluating the work of scientists in validating the Watson and Crick model of DNA replication. All this first part we've already looked at in our last lesson. Okay, so we're going to start off with an overview of the Watson and Crick model of DNA replication. Okay, so Watson and Crick discovered the structure of DNA in 1953, helped by Rosalind Franklin's research in X-ray diffraction. You don't need to know that second part, but you do need to know the first part. You don't need to know the date, but you do need to know that Watson and Crick were the people that discovered the structure of DNA. Okay. They hypothesized that DNA must replicate either conservatively or semi-conservatively. And the semi-conservatively part was the one that we looked at in our last lesson. Okay, so the semi-conservative replication of DNA, let's say we had a DNA like this. We drew loads of these in our last lesson. We saw that when DNA replicates, one of them acts as a template, one of them will separate, and then the other one will separate. And then after that, the process of DNA replication will actually start. Okay, so in every daughter strand of DNA, you will always have one original strand of DNA. Okay, so that's what we actually saw when we when we sort of looked at the replication of DNA. Okay, so it's either the conservative replication of DNA or the semi-conservative replication of DNA. So if semi-conservative is this way, when one original strand of DNA is conserved, so one original strand of DNA is conserved, then the conservative replication of DNA is when DNA is created from scratch. So if we have this as our DNA, we don't need to split this DNA into two separate parts and then they will create their own strand. It's not like that. It's basically from this DNA, we just create another one from scratch. Okay, we don't need to use the original strand of DNA to make the new strand of DNA. It's literally just that we use it as sort of from scratch. We're building something new from scratch. Okay. And then Mathelson and Stahl conducted an experiment which proved that DNA replication must be semi-conservative. So Watson and Crick were the people that discovered the structure of DNA, and they hypothesized that there were two possibilities of how DNA could replicate. It was either conservatively, when the DNA is created from scratch, or it could be semi-conservatively, when one original strand of DNA is conserved, basically. Okay, and Meselson and Stahl were the two people that conducted an experiment to get an answer basically, and they proved that DNA replication should be semi-conservative. Okay, which is the way that we know it now, about how DNA helicase breaks the adjacent hydrogen bonds. Each DNA sort of strand acts as a template. DNA polymerase catalyzes the formation of these phosphodiester bonds, and all the process that we looked at in our last lesson. Okay, that's the process. We know it because of these two people. Michelson and Stoll basically proved that this DNA replication was not conservative, it was semi-conservative. Okay, so that's just an overview of how DNA, what this sort of experiment is about and why was it done. Okay, because there were two scientists that discovered the structure of DNA, and they hypothesized that there was two possibilities of DNA replication. And then the scientists, students, Messelson and Stoll, they conducted an experiment to prove and get an answer that DNA replication was semi-conservative. Okay, so there were two hypotheses, okay, and we've looked at both of the hypotheses. One was that semi-conservative replication. So when DNA was replicating, it would replicate semi-conservatively. And what they meant by that was that each replicated DNA molecule contains one of the original DNA strands and one newly synthesized strand. So basically the semi-conservative replication of DNA that we know today. Okay, so if we had DNA like this, the first hypothesis was that this DNA would split into two separate strands of DNA like this and like this. It would just split apart. And then these ones will go on to replicate and form more DNA. Okay, each DNA will have its original parent strand. Okay, one original strand of DNA. Okay, and that was a semi-conservative replication of DNA. As you can see in both cases, 50% of the DNA is the original DNA. Okay, so it contains 50% of original DNA. Okay, there's always one strand which has been obtained from the parent strand. Okay, so that was hypothesis one. There were two possibilities of what it could have been, and this was the first possibility. Okay, the second possibility was that the original DNA remains intact following DNA replication, and then two newly synthesized strands of DNA will join together, and each time you will fully conserve the original amount of DNA. Okay, so we had the fact that we've already looked at this, but this was the original strand of DNA. Okay, this was original strand of DNA, and then basically in no relation to the original strand, it will just act there as the DNA. A newly synthesized strand would come about. Okay, like that. This is newly synthesized. Okay, there's no need for sort of splitting and acting as a template. All that would happen is that you would have the original strand, and then a fully new synthesized strand would come about. And that was the second hypothesis. They didn't know which one was right, but they made two hypotheses that these are the two possibilities of what could happen. And Watson and Crick students, Messelson and Stoll, they conducted experiments to show which one it was. Okay, and they came to the conclusion that it was a semi-conservative replication of DNA, and it was not the conservative replication of DNA. Okay, as you can see over here, the original DNA, we had 100% of the original DNA, and over here we have 0% of the original DNA. We are conserving all this 100% of DNA whenever the DNA is replicating. Okay, we're not using it to replicate a new strand of DNA. Okay, now this is basically the experiment. Okay, and this is one of the sort of cycles that I made. Okay, now there's a key part over here that you need to know, and that is centrifugation. Okay, and centrifugation you literally learn in topic 2. We've actually not gone to it. However, this experiment included the use of a centrifuge, and all a centrifuge does, it separates down sort of molecules according to density. Okay, so that's what this keyword over here, centrifuge, basically separates separates DNA strands slash molecules according to density. Okay, and basically what this um, experiment was basically you have a solution of bacteria, okay, and you have bacteria, and then you sort of you add different isotopes of nitrogen, okay, um, and the bacteria will take in the nitrogen to make new DNA, and then you would see how much of that specific DNA has been made. Okay, so let's say that in a solution, you would have a beaker, and in this solution, you would have bacteria, okay, and you would have the nitrogen isotope. Okay, and there are two nitrogen isotopes that we're going to be using, and that is the nitrogen 14 isotope and nitrogen 15 isotope. And basically what's going to be happening is that the bacteria is going to take in the 14 nitrogen and the 15 nitrogen, and they are going to basically create DNA. Okay, they're going to create DNA, and then we're going to use a centrifuge to see what sort of DNA they've created. Okay, the DNA created from the 14 nitrogen is basically going to be lighter because it has a lower number of neutrons. And the DNA created from the 15 nitrogen is going to be heavier. Okay, so the bacteria are going to be present in the solution. We're going to incubate the solution with either the 14 nitrogen isotope or the 15 nitrogen isotope. And then the bacteria are going to take in the nitrogen isotope, whatever we incubate it with, and they're going to create DNA. Some of the DNA is going to be lighter because they're going to be created from 14 nitrogen, and some of the DNA will be heavier because it would have been created from the 15 nitrogen. Okay, and the sort of centrifuge basically measures the density. Okay, so um, if you're using a centrifuge, the 14 nitrogen will come up at the bottom. So normally in a centrifuge, you will have to put in a test tube with the solution in. Okay, and if you're incubating with the 14 nitrogen, you will see a line come up towards the bottom because it's lighter. If you're using 15 nitrogen, you would see a line coming much higher because it's heavy. Sorry, it's much lighter. So if the line comes to the bottom, it's heavier. If the line comes to the top, it's lighter. Okay, and this will make sense if we go through this. Okay, so we had so in our first generation, we have bacteria grown in medium of 15N. Okay, so this is the heavy nitrogen. Okay, so all we have is we have a beaker, we have bacteria in here, and we add in 15N. So the nitrogen has a mass number of 15. Okay. And basically, once we've added in the 15 nitrogen, the bacteria will take in this 15 nitrogen, and all they will do is they will basically create DNA. Okay, so the DNA that's going to be created is going to be heavy. Okay, it's going to be heavy because it's going to contain 15 nitrogen and 15 nitrogen. Because remember, DNA comes in pairs, there are two strands, so each strand of DNA will have 15N and 15N. Okay, so when we centrifuge it, when we're centrifuging it, we pour the solution into sort of a test tube. Okay, once we've left it for enough time, and we centrifuge it at a high speed, and we would see that a line will appear at the bottom. And the reason why the line would appear at the bottom because there's only one isotope, there's only one type of nitrogen in it, and that nitrogen has only one density. Okay, it's really heavy because it's 15 nitrogen and not the 14 nitrogen. Okay, so I've drawn the heavy in pink over here, two lines. Okay, because remember it's a double helix, it needs two strands. Now the same solution, the same DNA that we have, we incubate it for another generation. And this time we don't add in 15 nitrogen, we add in 14 nitrogen. Okay, we add in 14 nitrogen. Okay, so to this solution now, what we're going to do is we're going to add in 14 nitrogen, and we're going to, the original 15 nitrogen is already in the solution, and there's still going to be bacteria in the solution. Okay, so we originally had this sort of heavy strand of DNA. We've just added in 14 nitrogen now. Okay, and when we've added in 14 nitrogen, the 15 nitrogen has each sort of split off and it has paired with a 14 nitrogen. Okay, so what they noticed was that we had a 15 nitrogen paired with a 14 nitrogen. Okay, if DNA replication was conservative, this is what would have happened. We would have had two pink strands over here, and then we would have two lighter strands over here, which were green. Okay, but that didn't happen. Okay, what happened is the heavier strand, the pink and the green paired together, the pink and the green paired together. The original strand of DNA, which was heavy, paired, sort of split off and paired with a lighter strand of DNA. Okay, so 100% of the DNA is 15N, 14N. And as you can see, the line which I've drawn over here after centrifugation is a bit higher than the line over here. Well, it's supposed to be a bit higher. Now, once we've done it for another generation, we're still doing with 14N. Okay, so when we're doing with 14N, we've just added more 14N into the beaker, 14 nitrogen. Once we've added in 14 nitrogen, what happened is that these two sort of DNA strands that we have, they split off even more. Okay, and this time we produce double the number of DNA strands. And this time, what's basically happened is that we get two lines on our centrifuge, because over here we have, remember, green is for lighter DNA. We have the lighter DNA, and then we have the heavier DNA, 15N and 14N over here. So 50% of the DNA is 14N, 14N, and then 50% of the DNA is 15N, 14N. Okay, because this is the 15N, 14N DNA because it contains a pink one, and this is the same, so 15, 14, but this is the 14, 14 nDNA. Okay, 14, 14 nDNA. Okay, because it's the lighter, and that's why there's two lines appeared on the centrifuge, because there's a 50% split between the heavy DNA and the light DNA. The 14, 14 DNA is lighter than the 15, 14 DNA. Okay, now over here, now we grow it in another medium of 14N. And this time the same thing happens. The soft strand split and the 14N is incorporated. Another green line is incorporated. And this time 25% of the DNA is 15N, 14N. This is the 15NN, and this is the 14N, 15N, 14N, 15N, 14N. And this is the heavier DNA, because it contains the 15N, and that's why, because you only have 25% of it, we have a thin line over here on the centrifuge. Over here, these are the lighter DNA. Okay, these are the lighter DNA, but we have a lot of them. That's why there's a thick line of lighter DNA. So thick line of lighter DNA. Okay, and that's basically the Watson and Crick model, what Messelson and Stahl experimented. Okay, they basically showed that DNA strands will always split when they are replicating. They will not replicate from scratch. If they replicated from scratch, this would happen where we've crossed out. We would get two heavy strands and then two light strands of DNA. Okay, if the sort of DNA replication was conservative, but it's not conservative, it's semi-conservative, and that's shown by the heavy strands of DNA, the heavy nitrogen strands of DNA have separated, and then each of them have been paired with whatever isotope of nitrogen they've grown in the second generation. So as you can see, we started off with 15N nitrogen, and then we've always used 14N, 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 14N. So every time the strands have separated, they have paired up with the new incubation of isotope of nitrogen, basically. And the purpose of this centrifugation is to show that you have different isotopes of nitrogen present in solution. When you have 100% of the same DNA, you will only have one line. However, when there's a mixture of DNA, for example, a 50-50% of split, there's two different lines shown on the centrifuge, one showing a heavier line and one showing a lighter line. Okay, and if there's sort of a 75% split to a 25% split, you will see a thicker line compared to a lighter line. Okay, and that is basically Messelson and Stahl's experiment. If you have any questions, please do let me know in the Q&A, so I can answer them. Just let me know in the Q&A and I'll be more than happy to answer them. Okay, I'm just going to pause there for 30 seconds for anyone to note down, and then we will move on. Okay, so hopefully you've had an opportunity to note that down. We're going to move on now. So basically, we're just going to sort of finish off the content. There's a couple of slides. There's, I think, 2 slides or 2 to 3 slides on quick notes. But we've looked at the process now. The centrifugation process, the process of growing the bacteria with different isotopes of nitrogen. First of all, heavier nitrogen, and then we move on to lighter 14N nitrogen. And then we saw that if DNA replication was fully conservative, then we would not sort of get the 100% of DNA, which is the exact same in the first generation over here. We only got it because the two strands of original DNA separated and the 14N paired with the original strands of DNA. And we saw that continuously. We didn't get the expected ratios, and that's why we proved that DNA replication was semi-conservative. Okay, now why did we use nitrogen? Okay, that's probably a question ringing in your head. We didn't use nitrogen because we did use nitrogen because DNA bases are nitrogenous. They contain nitrogen. That's why we use nitrogen, because it was a very accurate representation of what our DNA actually does when it's going through DNA replication. Okay, nitrogen has two isotopes, 14N and 15N. 14N is slightly lighter because it has a lower number of neutrons, and 15N is slightly heavier because it has one more neutron than 14N. Bacteria take in the nitrogen isotopes to make new DNA. Okay, that's why we had bacteria and we incubated it with different isotopes of nitrogen, because the bacteria is able to take in the nitrogen isotopes to produce new DNA. Okay. Bacteria which is grown in a medium containing 14 nitrogen will have DNA which only contains this isotope and will be lighter. Okay, so if you have bacteria and then you just add in 14 nitrogen, what will happen is basically the 14 nitrogen is what's going to be taken up by the bacteria, and you will only produce DNA which will have 14 nitrogen present, and that's lighter than 15N nitrogen. Bacteria grown in a medium containing 15 nitrogen will have DNA which only contains this isotope, and it will be heavier. DNA samples can be spun in a centrifuge to confirm density. Okay, so the purpose of a centrifuge, there is a whole lesson on the centrifuge, but basically the centrifuge is spun to confirm the density of the DNA. It's to confirm that different DNA, there are different components of DNA. Some parts of the DNA are heavier compared to other parts. That's why some parts will be sort of condensed much lower than other parts. If something is more dense, it will sink to the ground, and some of the components will be lighter, which means that they will stay towards the top. The denser the DNA, the more that it will settle lower, and the less dense DNA will settle higher. Okay, so the more dense DNA, which is N15, will settle lower, and the less dense DNA will settle much higher, which is the N14 DNA. Okay, and just a quick summary. Michelson and Stahl's experiment proves Watson and Crick's semi-conservative hypothesis. So remember, these were the people that hypothesized that DNA replication can either be semi-conservative or conservative. However, they never got to an answer. And then these scientists, Students, Michelson and Stahl, conducted the experiment to come to the conclusion that DNA replication is semi-conservative. The experiment involves growing bacteria in mediums containing two different isotopes of nitrogen. We used 14 nitrogen and we used 15 nitrogen, two different isotopes of nitrogen, one which is heavier than the other. The nitrogen isotopes are incorporated in the DNA nucleotides to make more nitrogenous bases. Okay, we've already looked at that. And then DNA is separated according to its density using a centrifuge. Okay, centrifuge is there to confirm density. It's to separate components of DNA according to their density. And if you spin them at faster speeds, you will see more sort of dense material being collected at the bottom of the test tube.